Why should you care what I have to say? I can explain. My name is Michael Wayne Clark. I have three names not because I'm pompous, it's because Michael and Clark are very common names for my generation, and the Wayne is a distinguishing factor. I got my PhD from UCLA, that's the University of Southern California at Los Angeles, in biology. And I did that in about the latter part of the 20th century, so yeah, I'm a bit old. I'm here now in Santa Monica in front of the ocean, as you can see, and I thought this is a nice place to introduce my video series, which is on helping to explain the fundamental concepts of science and biology, which I call I Can Explain. The reason that you should care about what I have to say is that I've been teaching these concepts for the last 30-odd years, and I know what I'm doing. Now to my experience. My PhD thesis was on the molecular structure of the ribosome. The ribosome is the cellular organelle responsible for protein synthesis in all cells. The organism that I worked on was a bacterium that is isolated from the human gut, called Escherichia coli, or E. coli for short. The species named coli actually is derived from where exactly it's extracted from, the colon. And again, that's the end of the human intestine and not the punctuation mark. Actually, you were already familiar with one of the main features of this organism, E. coli. One of the reasons that your flatulence smells, that when you pass gas and it stinks, it's because of a bacterial waste product. Yes, my lab did smell just like a big stinky. So what I did in my own defense was I always used to say, and I still do say this, is that don't blame me, blame the bacteria. Bacterium are generally categorized as prokaryotes, which means that they don't have a membrane-bound nucleus internally inside their cells. They're different from, say, like us, a cell which is referred to as a eukaryote, because a eukaryote has a membrane-bound nucleus within the cell. So once I graduated from UCLA, I wanted to do a couple of things. One, I wanted to move up the phylogenetic tree and study some cells more like us, and also I wanted something that smelled more pleasant than bacteria. That organism of choice was the yeast Saccharomyces cerevisiae, or baker's yeast. It fulfills both of those criteria. One is that it is a eukaryotic cell. It has membrane-bound nucleus. It is much like us human beings, and it smells much, much better. Okay? It makes the lab smell like baking bread instead of a toilet. Okay? Another good aspect of dealing with Saccharomyces cerevisiae, or S. cerevisiae for short, okay, is the fact that you can treat it much like bacteria, in the fact that you can grow them up in a relatively short time, and you can get billions of them, which you can kill without any guilt. To study S. cerevisiae, I took a postdoctoral fellowship at California Institute of Technology in Pasadena, California. At Caltech, I specifically studied how the genetic information, the DNA that's stored in that membrane-bound nucleus, got processed to be utilized in the cytoplasm to make proteins on, as I've spoken before, the ribosome, the cellular ribosome. I was at Caltech for 5 years studying these processes, and after that, I got an assistant professorship at McGill University in Montreal, where I had my own lab, and I further studied the eukaryotic nucleus using yeast, but in this case, I was studying the genes that were responsible for the actual structure and function of the nucleus. In my own lab, I was able to identify 5 genes that were involved in these processes, but were also involved not only in yeast, but also in humans, and the human genes could actually be utilized in yeast to cause a functional cell. So After 25 years of research in the lab, in academics, I decided to move on into business, but I still liked teaching. So I have been teaching part-time off and on in the various universities here in Southern California. And in the process, I've developed this style or method of teaching, which many of the students have found not only entertaining, but also very useful in the fact that they learn things. And so in these episodes of I Can Explain, I'm going to use those methods to help you understand some of the basic fundamental concepts of biology and science, and hopefully you'll learn something.